So in today's class I will take up glycolysis. So first we will be talking about what glycolysis is. Glycolysis, as it says, it means breakdown, glycolysis, breakdown of glucose. So this is a 10-step metabolic pathway. What happens in this is glucose is broken down into glucose is a 6-carbon compound which is broken down into 3-carbon compound known as pyruvate, and 2 molecules of pyruvate are formed from 1 molecule of glucose. Now we are going into some nitty-gritties of glycolysis. So it is a 10-step metabolic pathway where glucose is converted to pyruvate through a series of enzyme catalyzed reaction. So all these reactions are enzyme catalyzed, and glycolysis takes place in the cytoplasm of the cell. It doesn't take place in the mitochondria or nucleus. It's a totally cytosolic process. All the enzymes needed for glycolysis are present in the cytoplasm of the cell, and you get 2 ATP molecules produced in the end, net 2 ATP molecules, 2 NADH and 2 pyruvate molecules per glucose molecule. It's interesting that you actually 4 glucose molecules are formed, but 2 is utilized in the glycolysis. So net gain is 2 ATP molecules, and it's a universal process. It takes place in all the organisms present, from plants to animals to bacteria. This glycolysis is a universal process. Extremely important biologically. It is, first thing is that you need its primary ATP source in all organisms. So glycolysis produces energy. The next thing is it's a metabolic integration point. It's a central hub connecting major pathways. It connects into the Krebs cycle, which is even more central, and it has got outlets into the pentose phosphate pathway. It has got outlet into glycogen metabolism, gluconeogenesis. There are n number of pathways, and via Krebs cycle possibly all the metabolic cycles of the body are integrated into glycolysis. But Krebs cycle is the central metabolic pathway, but this is also another central metabolic pathway, but more central is Krebs cycle. And it is a biosynthetic precursors. That is, it provides building blocks for nucleotides, amino acid, for nucleotides via the pentose phosphate pathway, where riboses and deoxyriboses are formed, and amino acid also. There are through several of these amino acids can be synthesized from glucose through the different processes, and it has got a redox balance. It maintains the NAD plus NADH homeostasis. So NADH is it's a hydrogen carrier energy bond which is needed in the electron transport chain. So it maintains the amount of NAD plus and NADH in the cell in the right balance. NAD plus is already the hydrogen, it has been oxidized, the hydrogen has been removed, and when you reduce it, this NADH is a much more high energy form. This is generated during glycolysis. Now if you look at the small scheme of this glycolysis, the first part is the from the step 1 to the step 5 is the investment phase. Some ATP is invested, that is 2 ATPs are consumed in this process in the first phase, and that is very important. First thing is that it needs the glucose molecule which is there, it needs to be raised to a higher energy state for this reaction to take place initially, and so glucose becomes glucose 6 phosphate Glucose 6 phosphate has because of the phosphate bond, it has got a higher energy, and to give that energy is provided by a metabolically coupled ATP hydrolysis which is taking place. The second is the splitting phase. First investment paisa dala. The second is the splitting phase, where the 6 carbon glucose is broken into two compounds of 3 carbon. One is glyceraldehyde 3 phosphate, and which is an aldehyde, and a DHAP or dihydroxyacetone phosphate, which is a ketone. So essentially this dihydroxyacetone gets isomerized, converted into glyceraldehyde 3 phosphate. So from one glucose you get two glyceraldehyde 3 phosphates eventually. And now you have invested, you have done splitted. Now is the payoff phase from steps 6 to 10, where ATP and NADH is produced. ATP produced and NADH is produced, and the final product is pyruvate formation. So let us go out to the first step, glucose to glucose 6 phosphate. Glucose is the starting substrate, hexokinase is the enzyme which is needed, and it transfers phosphate from ATP. Hexokinase, so it takes the phosphate, the terminal phosphate of ATP, and it is attached, and glucose becomes a higher energy glucose 6 phosphate, and this enzyme is inhibited by glucose 6 phosphate. So if you give increasing amounts of glucose 6 phosphate, this enzyme is inhibited. This is known as substrate inhibition. That is, if there is lots of glucose 6 phosphate, then the body, the cell will know that okay, no more glucose 6 phosphate is not needed, so glucose don't consume ATP and become glucose 6 phosphate anymore. The second step is fructose 6 phosphate to fructose 1,6 bisphosphate. Again another ATP is utilized. So glucose 6 phosphate, in the presence of phosphoglucoisomerase, is converted into fructose 6 phosphate. So, and glucose 6 phosphate is in an aldehyde form. It becomes into a ketone form, that is fructose. Now phosphofructokinase is a regulatory enzyme which again utilizes one more ATP and forms fructose 1,6 bisphosphate. This phosphoglucose fructose is a very important regulatory enzyme in glycolysis, or it is the rate limiting step. It is allosterically regulated. Allosteric regulation means any regulation which is where the regulator binds to any site apart from the active site. It binds to another site, and by binding to another site, it causes a conformational change which infects or affects the activity of that particular enzyme. So here it is ATP. If the ATP amount is high, then there is no more glycolysis requirement. So because ATP is there, why do I have to spend glucose to make more ATP? So it is stop phosphofructokinase. So this ATP, excess ATP which is present, it binds to another site of the called the ATP binding site, and it inactivates the enzyme, and glycolysis stops over there. There are parts of this regulation which I will go into details later, and eventually you have even a higher energy for compound, that is fructose 1,6 bisphosphate. So PFK1 or phosphofructokinase 1 enzyme, it is the key control. So when there is high amount of AMP, that is adenosine monophosphate, So ATP has been broken down, hydrolyzed several times, ADP and then again AMP. If the AMP level goes high, that means it indicates that ATP is less. So the body needs more ATP, and so glycolysis start. So this phosphofructokinase positively makes this AMP and ADP positively regulates PFK1 by binding to its allosteric site, and the activity of PFK1 increases and more fructose 1,6-bisphosphate is produced. There is another compound known as fructose 2,6-bisphosphate. So what happens when fructose fructose 1,6-bisphosphate is accumulated, some of it gets converted into fructose 2,6-bisphosphate, and this inhibits PFK1. So lots of fructose 1,6-bisphosphate means that lots of fructose 2,6-bisphosphate, similarly the other way around, and this will stop the glycolysis reaction, means lots of product is present and the regulation is stopped. Negative is ATP, citrate, H+ ions, glucagon. So in a fasting condition, ATP is needed. Glucagon, what it does, it will stop it. Fasting condition, ATP is needed, but what happens is body tries to take its energy from non-carbohydrate sources through a process known as gluconeogenesis, that is production of glucose from non-carbohydrate sources like acetyl coenzyme produced from lipid metabolism, from protein is converted enters into the Krebs cycle, and this forms glucose. From protein you can make glucose from amino acids rather, and during the fasting stage, glucagon increases the blood sugar level through gluconeogenesis steps, not glycolysis. And insulin, what will it do? Insulin also decreases the blood sugar levels, and it stimulates breakdown of glucose. If people like me who are diabetic, for them there is low insulin amount as well as insulin receptors. So what happens? Our blood sugar level, blood glucose level rather, is very high. This is not desired for a good life. So you have to regulate your insulin. So you have to take insulin injections, and this sort of reduces blood sugar levels. It will promote breaking down of glucose into pyruvate, give you energy, and glucagon does the other way around. In step 4 to 5, there are 3 enzymes. One is aldolase. It cleaves fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This is a type of opposite of aldol condensation reaction, where aldehyde and ketone, in aldol condensation it happens at 2 aldehydes or one aldehyde and a ketone, they condense together and form one molecule. That is aldol condensation, and this aldol is broken. So basically fructose 1,6-bisphosphate is an aldol. It is broken down into dihydroxyacetone phosphate, which is a ketone, and a glyceraldehyde 3-phosphate. Then this dihydroxyacetone phosphate doesn't enter, it is converted back into glyceraldehyde 3-phosphate. So actually you have 2 molecules of glyceraldehyde 3-phosphate formed, and so this isomerization is done by the enzyme triose phosphate isomerase. Now this glyceraldehyde 3 phosphate continues into glycolysis. Now is the step where energy formation starts to take place. There is the very common enzyme glyceraldehyde 3 phosphate dehydrogenase, which is constitutively expressed in all cells. It doesn't increase, it doesn't decrease. This is perfect. So what it does, it oxidizes glyceraldehyde 3 phosphate and it produces 1,3-bisphosphoglycerate. And in this process, NADH is generated. It takes one hydrogen out from glyceraldehyde 3 phosphate and it gives it to NAD plus and forms NADH plus H plus. Next is phosphoglycerate kinase. It transfers phosphate. It produces 3-phosphoglycerate. That is, it was 1,3-bisphosphoglycerate. So one phosphate is now transferred to an ADP and it becomes 3-phosphoglycerate. One step down, the energy falls in 3-phosphoglycerate. It generates ATP. This is the first energy which has been yielded. So one molecule of 1,3-bisphosphoglycerate forms one molecule of ATP. And so two molecules, since there are two glyceraldehyde 3 phosphate molecules entering into the glycolysis, so it is multiplied by 2. So you generate 2 ATP molecules in this particular step. So per glyceraldehyde 3 molecule, 1 NADH is produced, 1 ATP is generated and a 3-phosphoglycerate is formed. Step 8 to 10 is the conversion of pyruvate. Enzyme is phosphoglycerate mutase. This converts 3-phosphoglycerate mutase into 2-phosphoglycerate by moving the phosphate group. The phosphate group was at the 3rd carbon. It goes to the 2nd carbon. Then enolase. What enolase does is it converts 2-phosphoglycerate to phosphoenol pyruvate PEP by removing water. Water is removed over here. And pyruvate kinase converts PEP to pyruvate. So phosphoenol pyruvate has got one phosphate. That phosphate is also removed and it is used to make ATP from another molecule of ADP. Since there were always two molecules present, as 2 glyceraldehyde 3 phosphate had entered, so this is also multiplied into 2. So eventually if you multiply it into 2, you have 2 ATPs formed in this step, 2 ATPs formed in the glyceraldehyde in the previous step, phosphoglycerate kinase step. So 2 plus 2, 4. But initially in the first investment phase, already you had already lost the 2 ATP. You had lost 2 ATP molecules. So net production is 2 ATP molecules. So pyruvate kinase is regulated allosterically. You know what allosteric regulation means. Fructose 1,6-bisphosphate binds to allosteric site, inactivating enzyme by stabilizing active conformation. Allosteric inhibition. ATP and alanine bind to inhibitory sites, causing conformational change that reduce activity. And you know hormonal regulation, which I told, glucagon. It actually stops glycolysis. Hexokinase and glucokinase isoenzyme regulation. So this is basically a comparison of hexokinase and glucokinase. Where it is located, the hexokinase is located in brain, most tissues, and glucokinase in liver, pancreas and cells. Hexokinase has got a much stronger affinity for glucose than glucokinase. That is because during times of starvation, brain gets the first call for glucose, not liver. So glucokinase's affinity is much lower. Now there is transcriptional control where insulin upregulates glycolytic enzymes, glucagon downregulates them, HIF-1 alpha induces enzymes during hypoxia. There is allosteric regulation, ATP by AMP ratio, it controls PFK1. Then you have post-translational modification also taking place on the at the protein level. And you have tissue specific regulation like glucokinase and hexokinase. Glycolysis dysregulation underlies numerous pathologies. Cancer cells exhibit the Warburg effect. Now what is this Warburg effect? So it has been seen that people who have get cancer, suddenly they become very thin, they lose tremendous abnormal amount of weight. Same with diabetes, there is a weight loss which is a symptom. What happens in this Warburg effect is cancer cells, they don't do oxidative phosphorylation, or they don't go into the mitochondria, they don't they can work without the presence of oxygen. So this particular phase, glycolysis, you don't need oxygen. But the next part actually, which is inside the mitochondria, the Krebs cycle and especially the electron transport chain, they require oxygen. So cancer cells, they can grow even in hypoxic conditions where oxygen is less. So it has been seen that these cancer cells, they start producing making their ATP only through glycolysis. There is no Krebs cycle and electron transport chain going on, or very less. So as you see that around 30 to 32 molecules of ATP are produced from one molecule of glucose during Krebs cycle, glycolysis and electron transport chain. You break down the rest of the 28 molecules of ATP, you get only net 2 ATP molecules. So to meet the ATP demand, the cells utilize up all the glucose source of in store, utilize up all the glucose in store. Result is the cells shrink in size, or people start making more and more glucose. Your fat, protein, they are all sort of broken down to make glucose for the cancer cells survival. So this is known as Warburg effect, that only it has been seen in cancer cell that they get their ATP only through glycolysis, no other process. And lots of glucose are taken up, glucose stores, fat stores, your protein stores, muscle breakdown, all starts to take place, and then you become thinner. Okay, there are 10 enzymatic steps, 3 regulatory points, 4 ATP, so 2 ATP net gain, and 30 plus therapeutic targets. Do you people have any questions? আচ্ছা, অ্যাটেন্ডেন্সটা দিয়েছো?
Yes sir.
ঠিক আছে, দুদিনের দিয়েছিস তো? দুবার দিয়ে দিবি?
Yes sir, দিয়েছি।
হ্যাঁ, দুবার দিয়ে দিবি, ঠিক আছে। ঠিক আছে, বাই বাই। তাহলে আবার আমি পরশুদিন